My name is Cara Fiore. I'm an assistant professor in biology here at Appalachian State University. And I'm a marine microbiologist, so that means I ask questions about microbial physiology of marine microbes. I also ask questions on how microbes influence nutrient cycling in the ocean, and questions about coral reef ecology and microbial symbiosis as part of that coral reef ecology. The sponge microbial symbiosis, when I use that term, that really refers to any interaction, whether that's beneficial or some other interaction of these microbial symbionts, so beneficial or not. So my research has focused on these sponges and their microbial symbionts, asking questions about what shapes this microbial community, what kind of interactions are there between the sponge host and these microbial symbionts, and then as well as nutrient dynamics on coral reefs. So sponges are filter feeding animals, meaning that they draw water in and then they expel water through usually a central opening or multiple openings of that sponge. And they're filtering out bacteria that's in the water as well as some dissolved nutrients in the seawater. And so if we think about sponges, a lot of sponges on a coral reef, you could imagine that you could have a pretty big impact just from that filtration capacity of those sponges and whatever they're taking out of the seawater or putting into the water. So my research on the sponge microbe system has really focused on kind of the microbial symbiosis aspect as well as this nutrient dynamic aspect. So I was fortunate to go to a workshop as a master's student when I started working on sponges, sort of by chance. And there at this workshop, I met some really amazing mentors. So people who were really excited about sponges for one, and I thought this is pretty fun. And this is pretty fascinating. And those mentors that I met at that workshop are people that I continue to work with today. And one of the things that really fascinated me are really some of the aspects that I continue to focus on today. So the ability of sponges to filter this vast amount of water and what that might mean for the coral reef environment. So at the time, even though a more prevalent and kind of popular idea is looking at corals, which are an important aspect of coral reefs, they're the reef builders, but the idea that these sponges that we don't really know a whole lot about this group could impact these large-scale processes of nutrient dynamics on coral reefs was really fascinating to me. Also, these are what we call the most ancient living metazoans. So metazoans just refers to multicellular animals. So this group of the sponges, these are our kind of most ancient living ancestor to all multicellular animals. They don't have what we'd call a gut. They don't have true kind of tissue layers like we see in other animals, but they do have specialized cells. They do have specialized functions. And despite being able to filter vast amounts of water, taking in these bacteria for food, they're somehow able to maintain these distinct microbial communities. And so how are they doing that? These group of specialized cells in a way. So these aspects were really fascinating to me, and one of the reasons that I continued to work with them really from that point on. One of the exciting things is that we've seen that we do in fact see differences in how sponges and their microbial communities can process nutrients that are dissolved in the seawater. So if you think about dissolving some sugar in water, that would be an example of this dissolved nutrient. And in seawater, there are components, dissolved nutrients from simple sugars to amino acids, so things that are the building blocks of proteins, and then much more complex molecules that are dissolved in the ocean that we don't really know what they are. But we do know that this dissolved nutrient profile is an important nutrient source for sponges. We don't know how much of that is facilitated by the microbial communities of these sponges and how that may differ across sponge species. So we know that the sponge species tend to have species-specific microbial community. And so one of the things that has been really exciting that we see is that we find that we do in fact see this different nutrient profile across sponge species. So these sponges are taking particulate components and some of those dissolved nutrients, pulling from those different resources differently across species. One of the things that we've been interested in is, is this in part facilitated by the microbial community? So if we think about sponges on a coral reef, if we've got, say, 50 sponges all living together in an area, one of the questions we had is, are they all doing the same thing? Are they all filtering the same particles and maybe the same set of dissolved nutrients? Or if you've got these coexisting species, have they done some sort of, is there resource partitioning that the microbial symbionts may be facilitating? And so we're starting to see that, yes, it looks like we see differences across species. So that may be some evidence for resource partitioning and some role of the microbial symbionts of these sponges. And this has really important implications in thinking about the structure and assemblage that we see on coral reefs. So if these microbial communities have sort of influenced the trajectory of sponges, what we see currently on coral reefs, that would be some of the first evidence to really show that. So to assess the impact of these different sponge species with their different microbial communities on the nutrient profile on coral reefs, we were able to collect the seawater going into the sponge and then the seawater coming out of the sponge so that we could compare that nutrient profile. So we did this by either collecting that water with a syringe or by setting up this setup we had with bottles that were pressurized and connected to a line that we could place over the opening of the sponge to collect that water that's released from the sponge, or right next to it to collect the water that would be going into the sponge. So we did this setup across sponge species and look at what dissolved nutrient components were taken out by the sponge or added in, what bacterial cells or other cells that were removed by the sponge potentially for food. And once we know then that we see these differences in this resource use, our question was then, what are the important nutrients or particulates like bacteria that are important to these differences that we see across species? What's accounting for a major part of those differences? And what does this mean at an ecosystem level? So what does this mean for a coral reef ecosystem? So what we found is that there is indeed an impact of the sponge community producing certain compounds that changes the nutrient profile of the coral reef at an ecosystem level. So these are some of the first data to be able to show this. And we could see that there are particular nutrients that we can identify. So certain sugars, certain amino acids or derivatives of those. So these things that we call metabolites. So amino acids, vitamins, and similar compounds that organisms use for growth. So we could see that sponges are consuming some of these particular nutrients and they're releasing some of these particular nutrients. And this is really important because they're releasing some certain nutrients that other organisms are using for growth. Nutrient profile can influence the free-living microbial communities. There are microbes just living in seawater. And those microbes are an important food source for some sponges and for other organisms. But those free-living microbes, you can also have what we kind of consider a healthy microbial community in the seawater. And then also a community that may have what we'd call opportunistic pathogens. And those can be particularly detrimental when it comes to corals and coral disease. So we've recently seen coral disease in the last few years move through the Caribbean. And this is really detrimental to these reefs, to coastal communities that rely on these reefs. Some of the work that we see, we have uncovered a connection between what these sponges are producing and how that may influence that overlying microbial community in the seawater. So is that going to drive more of a healthy kind of microbial community assemblage in the seawater, or one that has more opportunistic pathogens? And that's what we don't know yet. So some of our work is starting to uncover that for the first time. I hope that this work will be a seed for more community-focused work on coral reefs, and specifically that we'll think more about sponges as an integral component to coral reefs, which so far they have not largely been part of the community when we think about the community or ecosystem level of coral reefs. Sponges are often not really included in that. And in part, that's because we don't know a lot about what we're doing. And that's why some of this work is so important. So it's going to be critical that we still have organism-focused work. So asking questions about a particular organism, its role on coral reefs, its ecology, its physiology. But I want to see more studies that will include, when we're asking questions about coral reef ecosystem, a response to some environmental stressor that has happened perhaps, that will include sponges in this ecosystem consideration. We've also started to uncover connections between sponges and corals on the reef. So nutrients that sponges are releasing may be taken up by corals. And so there are some implications here for thinking about coral restoration. So when we do coral restoration, we're typically raising small fragments of a colony into larger fragments and then hopefully into these larger colonies. But are there implications for thinking about what kind of maybe assemblage that those corals could be on to support their growth? Maybe there's some role for sponges in that really hasn't been considered before.